thank you for the invitation in the first place. I was very flattered. Reproduction is a topic that is very near and dear to my heart. And I have over the years learned to really have a good appreciation for Xenarthra. So this was like a great excuse to go spend hours and hours reading about all of this stuff. What I'm going to talk to you guys about a little bit is some personal experience. Obviously, there's a lot more out there than what I have done. What I have done is mostly with animals under human care and in a partnership with Instituto Tamandua that Flavia Miranda has been kind enough to partner with us. So I'm very happy with that. But other than that, it's really just an appreciation and a recognition of all the work that other people have done. And so I tried to summarize and kind of organize some of the information that is out there. This is my email. If I missed your paper, if you know of something really cool that I did not put in my talk, it was not intentional. So please let me know because I hate that I missed it. Feel free to send me the paper, the information, whatever it is you have, because I would love to be made aware of anything that I wasn't. It's sometimes hard in the US to get all of the publications from Latin America because they're not all recognized here. So I am sure that there's a lot of stuff that my search engines didn't pick up. So if there's anything, any gaps that you see, please let me know. And with that, so the Xenarthrans occupy a huge range. And as you can see in this map, so much of it is in South America. There's some, in the US up here. Can you guys see my arrow over here? So you can see up here, it's probably mostly armadillos. And then in Mexico, there are some tamandras, but really the vast majority of all the diversity is going to be in South America. So they are mostly organized in a solitary or in pairs, some family groups, but they really don't function in big herds like other groups of species do. We have the anteaters, which are terrestrial, as well as the armadillos, and then we have some that are arboreal or that like the trees mostly, but also can come onto the ground. And with all of that, obviously all of their mating and rearing strategies are affected. Here's a little bit of the taxonomy. There's really such an amount of like that diversity, so many species that it is understandably a gargantuan task to even try to wrangle all of this information. So I'm hoping that what I was able to accomplish with this is just give you guys some idea of the different options that are out there within this group, as well as some of the gaps that we still have, so that we can focus new research on all of this. We have the sloths up there, the anteaters, and then a ton of armadillos. There are many places in the US where people don't really like armadillos. I don't understand that. I think they're amazing animals. The other ones have a little bit more of a charm factor and are easier to do some publicity for, but I really feel that armadillos are undervalued. Cool. So the Edentata have, I'm fascinated by their class. We have one species of giant anteater, 2 of the lesser anteater, and now 7 that we know of the silky anteater. Of the 2-toed sloths, there's 2 species, and of the 3-toed sloths, there's 4. And my personal ones are the chocolate ones. I know this is probably a little bit of a cliche, but I really think it's funny how much they look like chocolate croissants because I do love both sloths and chocolate croissants. So please indulge me in my little bit of humor here. Armadillos, the word itself comes from Nahuatl, which means turtle rabbit, and it has to do with, obviously, their appearance. There are 9 genera and 21 species, and they have a huge range in sizes, as you can see by these 2 species here, the giant armadillo and the little fairy armadillo. Moving on into reproduction, we're going to start with a male. They have intra-abdominal testes, all of them. The anteaters have them just caudal to the kidneys, and their size does vary a little bit in animals that are seasonal. There are many animals that are not, as we will see in some of this reproduction, that are seasonal in the sense that there are peaks in births, but not because there's a season where they don't reproduce. So depending on how you define birth season, some of them will be called non-seasonal because there's births throughout the year, but there are distinct peaks in the frequency of births. Externally, they have a penis. The tamandra has a penis that lacks a glans. It is conical shaped and kind of short. It is pointed backwards and it is fibroelastic. They do not have a baculum. Internally, they have the accessory sex glands. They do have the testes internally, as I had mentioned, and they are so close to the medial line that the epididymes are in contact. And this is important to know anatomically if you're going to be ultrasounding them and doing some of these measurements. In armadillos, the testes are a little bit more caudal and they're almost inguinal. Some people have hypothesized that they are internal because it helps them maintain that temperature. They have a lower metabolic rate, so the temperature tends to be a little bit lower than in other mammals, which the theory there is that the testes are external in order to keep them cooler than their core temperature. And in these guys, they wouldn't need to be that way. So their core temperature is a little bit lower, which means that they can keep the testes internally and a little bit better protected. They do have a prostate, which is bilobed. They have seminal vesicles and they have bulbourethral glands. The bulbourethral glands have been documented as present in all three groups of anteaters. In the 9-banded armadillo, they have been documented to be missing in the sloths, the C. hoffmanni and bradypus species. There's a lot of species within the Xenarthrans for which the anatomical studies have not been carried out. So some of these features have not been documented to be either present or absent. In the southern tamandra, this is what on necropsy it looks like. And you can see here the kidneys, and then right below are the testes. In this picture on the left, the Ks are the kidneys, and this white T are the testes. On ultrasound, they look like this. Here on the right, we see the ultrasound image, and you can see that the testes are literally like right up against each other. One of them looks a little smaller than the other one because they're not at the same level. So as you're scanning from cranial to caudally, one of them will appear first, and then it'll be a little bit bigger, and then it'll disappear, and then the other one will follow the same pattern. So if you're going to be measuring the widest part of the testes, it will not be in the same image because they'll be in different planes. The active spermatogenesis, like in any other species, if there is active spermatogenesis, the testicular size will be bigger because there'll be just more happening. There'll be more cells in there. In the giant anteaters, similar stuff. We have the kidneys here, and then just caudal to them. We have the testes, same thing. You can see the epididymes really in contact with each other. They're really centrally or medially located right there. They do have a persistent Mullerian duct structure that was documented, although the etiology and the function are unknown. It may just be a remnant that there was no evolutionary pressure to get rid of. And that is this pink one here. In the Silky anteater, there are no papers out there, although I did have the privilege of ultrasounding a couple of them, and here's a video of it. And you can see the kidneys here, and then as we move into a caudal direction, you can see the testes showing up there also in different planes, and then the bladder right there. So it'll be the same thing. You can start kind of right under the xiphoid and move caudally, and you will come across the kidneys, and right caudal to it the testes will appear. The sloths, like the anteaters, have small external genitalia, and the sexes can be difficult to correctly identify. The testes are internals, contributing further to this. In the US, there's actually been a couple of instances where animals have been misidentified in their sex. And so there's been, why aren't they breeding? Well, because they're both males, or they're both females. I was involved also in a case where one of the, a pair was not breeding, and they thought to check genetically, and then it came back that they were both males, and they thought that that explained why they weren't breeding. However, then we did an exam, and on x-rays, one of them was pregnant, proving that it was a female, and they just hadn't gotten around to it. But this means that sometimes, even depending on the genetic testing that you have, and how much has been done with them. If they don't have the right primers and stuff, there may be some errors there. So even the genetic testing is not infallible. For armadillos, they, again, have intra-abdominal testes, although they are more caudal than the anteaters. And I think that that is the part where you can't technically call them testicondi anymore. I removed that term from my presentation because I thought when I started to write this, I thought I knew exactly what it meant. And as I was reading more, I wasn't quite sure what the subtleties were. So I didn't want to use it incorrectly. In the armadillos, the testes are not joined to one another in the papers that I was able to find. True histological glance has been described for one hairy armadillo, but in general, they are not a group that is thought of as having histologically identifiable glands. They do have a seasonality in spermatogenesis. Some of them are continuous, and some of them have periods of rest and rut. So it is important to document it in the species, but also potentially for the region, because there are some species in which there are contradicting reports. And some of this may be just that we need more accurate data as technology evolves. Maybe it's, you know, the original data was just based off of births in the wild. In others may be that there is a certain adaptability on the environment. There are examples of other species where animals are seasonal or not seasonal, depending on the food availability, which may, you know, South America is a huge piece of land that has a lot of variability in seasonality, ecologically, environmentally, as well as food-wise. So there may be reasons why the same animal is seasonal in one area, but not in another. So when you see some of this contradictory data, that may be one of the reasons. Unlike anteaters and sloths, armadillos do not have small external genitalia, except, you know, the testes, which are internal. And in fact, this paper here, they used the penile length for aging in the giant armadillo, which I thought was fascinating and a super interesting fact. And so these guys are definitely not hard to identify as male or female based on their external genitalia. They do have a bit of a variety in morphology of this external genitalia that is just species specific, but in all of them, they seem to be easily recognized. There are differences in sperm morphology. Sperm morphology is actually one of the topics that I did find a fair amount of literature in on all these species. They are all kind of, you know, the paddle shaped, although some of them have a little bit rounder shape and some of them have a more elongated shape. There are some differences in how long the acrosome envelope is, but they all have pretty much the same basic design. In looking for artificial reproductive techniques, since some of them are fairly endangered of these species, I tried to make a table, and there is the one that I found the most for is electroejaculation. There is a couple of them of semen collection where I found it in giant anteater and the Silky anteater. I may have missed a couple of them. I thought I remembered that they had done it in the tamando as well, but I couldn't put my hands on that paper again. And I have not seen pharmacological semen collection in any of these guys here. I did find one paper where they tried a urethral catheter, which is a really taking off as a popular method for semen collection in felids and other carnivores. It is nice from the point of view that it is very low tech. If you have a catheter and you know how deep you need to put it in, you don't really need a lot of batteries and electricity and other equipment. The downside of it is that you usually collect very little of it. The volumes are definitely much, much lower than with electroejaculation because you're not getting any of the accessory sex gland stimulation. And so it is a lot smaller, the volume, but the concentration is a lot higher. So depending on how one has gone about it, you can still get a fair amount. One of the things that I have done in cats is that you combine the urethral catheterization with a good anesthesia protocol. And so you're really kind of combining the two semen collection methods, right? The pharmacological and the catheter. The pharmacological in the sense that you're kind of increasing the amount of semen that you can get in the urethra, and then the urethral catheter to really get everything that is inside rather than just relying on the one 10 to 20 microliters drop that comes out of the urethral opening. I did not find any papers on gamete rescue, so post-mortem. And it may be that in many of these environments, post-mortem gamete rescue, certainly from the wild, would be a long, long shot, because usually we won't find these animals until they have sat in the sun for a long while and they are quite ripe. So that may be one, but it may definitely be an option for animals under human care that were not collected prior to death. It might be a very valuable tool for animals that have come in from the wild as a rescue and then did not survive for very long under human care, as that would provide a lot of gamete resources if they can be frozen. As far as females go, they have they don't have a true cervix, at least in the anteaters. It is just kind of like a constriction. And so it's hard to pinpoint exactly, other than histologically, where this starts. It does provide us with a great advantage when we're doing some other ART, and I will go into it in a little bit. This is giant anteaters. They have a very long vestibule and vagina, which is odd compared to the very short, relatively speaking, penis that the males have. So it is not really one of those species like, canids or rhinos that have a large amount of volume of the penis that then can put the semen right at the cervix, and then with volume and with pressure can just basically do a transcervical insemination. Here, the semen really has to swim the whole way. They do have persistent Wolffian ducts as well. We don't know the function. When I first started working with Xenarthran, I was really surprised to see that they have a simplex uterus. It is very similar to primates in a lot of ways, as far as their anatomy goes. In particular, New World primates, because they don't menstruate like Old World primates. So this is in the Silky anteater, in the Tamandua, in the giant anteater, the sloths, and some armadillos. Armadillos are the one group where there's a little bit more variability. Just like in primates, the ovaries are on either side of the uterus, which makes it a little easier to find them on ultrasound because they're kind of attached. And so if you can find the uterus, which is a little bit easier to find if you do have a bladder, because it'll be right dorsal to it. And then you can find it, the ovaries here on either side. Depending on the species, sometimes the oviducts are a little bit longer, so the ovaries may be a little bit farther from the uterus, not quite as attached, and a little bit harder to find. The three-banded armadillo also has a uterus simplex. This is from a ovariohysterectomy, so they didn't go very deep into the pelvis there, so it's kind of cut up here. But on ultrasound, you see it just like primates, and this is, this what would be the cervix. And so this is the longitudinal view. The abdomen is here at the top. The fundus of the uterus is to the left, and the cervix would be to the right. In the middle, you can see the lumen, what would be the endometrium there, and you can see that the myometrium is a very even, echogenically speaking, structure. As I was saying, the armadillos have a little bit more variability. There's some where the uterus is simplex and some where it is bicornuate. Some of these have a functional uterus simplex, which means that anatomically it's bicornuate, but the pregnancy is always in the body and not in the horns. So from a functional point of view, it operates as a simplex, and I don't know, maybe evolutionarily that's how it came to be as well. Okay, so the brown-throated sloth, it has a uterus simplex with a long caudal segment, and we can see that here in yellow in this diagram. They have a urogenital sinus here, and they have two cervices, so they have a bipartite clitoris as well with paired crura. They have, in species where semen often needs to travel far, they have these folds that potentially can help semen traverse the long distance. Ovarian cycles is an area where there's a lot missing. Obviously, when you're doing field work and in the wild, samples are a lot harder to get regularly, and many of these species are not kept under human care. But even for the ones where they are kept in human care, there have been some challenges. And so the literature on this is quite sparse. In sloths, for example, fecal samples are a sample that is often used because it doesn't soak up into the dirt where these animals are. If they're boreal, urine samples are certainly hard to come by. But sloths defecate with such low frequency that it is hard to get the number of samples in to really get an accurate idea of the ovarian cycle, of what the hormones are doing. Because even though, they have a low metabolism, doesn't mean that their ovaries aren't progressing and that the estrogen isn't rising, within a number of days. This is a graph from Hay et al. From 1994 on Tamandos, where they did do urine. This was under human care, so they were able to get the urine. In this study, they also did vaginal cytology, which clearly you're not going to be doing if you're doing field work. They found that red blood cells appear 2 to 3 days before the peak in estrogen, and this graph shows those findings. They did estrogen, and then they plotted also these red blood cell peaks. And so they kind of overlap a little bit. And so really the presence of this blood is much more comparable to the pro-estrous vaginal bleeding that dogs have and not the menstrual bleeding that primates have. So it would probably be, I didn't have pictures of the vaginal smears, but in dogs, the blood would come in through diapedesis. So you'd be able to see actual erythrocytes. Whereas in primates, the blood comes from the degeneration, the sloughing of the endometrium, so you don't really see clean little red blood cells. They did look at the cornified epithelial cells, and it did not really seem to correlate with onset of estrus, so it doesn't seem that that would be a way to plot the estrus cycle like it is in dogs. It seems that looking for the red blood cells would be a much more efficient way to figuring out what the estrogen is. In this paper and in several others that I found where they did do some analysis of hormones, it seems that the people tend to analyze one, so estrogen or progesterone, but not both. And so if you're really going to try to do things like artificial insemination or any other more advanced assisted reproductive technique, it's important to have them both, because with the estrogen, you want to know how often they're cycling, how often they're coming into estrus, how often they're growing a follicle that reaches a mature size to be able to ovulate. And then you also want to know whether the ovulation is spontaneous or not, whether every estrogen cycle is followed by a luteal phase, or whether they're induced ovulators. I have not found, literature pointing one way or the other one definitively in a lot of the species. Pregnancies in this one, the 9-banded armadillos are probably the most famous of all there with their quadruplets. Most of them will have one offspring per pregnancy, and that matches the primate model. They have seen twins on occasion, but it is definitely a rare phenomenon. The 9-banded armadillo is a lab model for leprosy, so they've been studied in a lot of detail, probably much more than any other armadillo species for that reason. As far as monitoring pregnancy, fecal hormones can be one way. Tamanduas in particular seem to be very adaptable and open to be trained. Many of them are used as ambassador animals. Clearly, this is not the case if you're in the wild. But the ones that are under human care have served as a, good place to learn a lot of things, because you can ultrasound them and look at them on a regular basis, once or twice per week for a long period of time and really kind of follow growth. So that's what we did with that one. Her name is Rio. She was an amazing tamandua. And so it isn't hard to find the uterus, and you can see here the myometrium, the uterine wall, and then the developing embryo on the inside. And in this one, for example, here you can see the placenta up at the top, and we'll talk a little bit more about that in a little bit. And as the pregnancy progresses, the embryo obviously grows, but you start seeing other things like claws and ribs. And then eventually one day you find the skull, and it looks, exactly like what you imagine an anteater skull should look like. This is always a very rewarding view because it is very typical, and it is easy to get to know that you have the right view, shall we say, because it is you will have this round part to the back of the skull and then the long nose. And so if you can measure this biparietal diameter, we published this paper where we looked at the days before birth in order to estimate a due date. It is not really like the actual gestation length from ovulation. I haven't come across a paper where they documented, a couple of cycles and then estrogen and followed by the progesterone that doesn't come down, so that you can really kind of pinpoint when progesterone went up and when you can talk about a pregnancy, and then at the end of the pregnancy with birth when the progesterone drops. So that's kind of why we did it from birth back as opposed to gestational age forward. And what we found is we had, I think, like 15 of these pregnancies, and they were done by various different researchers across various different institutions with a lot of different equipment. And so several of them were, we did get an average, but they varied a lot. And so the biparietal diameter was one where they were all really pretty tight. And I think that it is due to the fact that it is a very recognizable view. And because you're measuring a calcified object, the skull, it is easy for people to take the same measurements, even if we're not trained by one and the same person. So it is a measurement that is very objective to find, and where it's easy to not have interpretation or other errors along with it. So if I was going to recommend anybody, I would definitely recommend this. Other measurements that we took were the skull length, the heart diameter, the chest diameter, the abdominal diameter, things like long bone length, which is commonly used in humans, are very hard to get exactly in the right plane and know that you have the full bone in view from end to end. So I find those a lot more variable in their reliability. This is a video that doesn't play appropriately, so I'm just going to show you but it's a sloth pregnancy. So we were able to see the chest here of the baby and the heartbeat inside. This is the one that I was telling you guys. We did find that we confirmed that the sloth was a female because we did find a baby inside. And by the time we found this baby, she was only like a couple of weeks away from giving birth. As far as placentas go, the anteater has a hemocorial and villus, so that puts them down here. And the hemocorial placentation is similar to primates as well. The discoid morphology of the placenta is also primate-like. Some primates have bidiscoid, just like some anteaters have bidiscoid. For example, in gibbons of the same species, some individuals can have one disc, whereas others will have two discs. That is not a species-specific thing necessarily. The tamandra has a hemocorial and discoid placenta, and as I showed you on those ultrasound images, you can actually image the placenta. If you don't know about this, I'm sure many of you guys do, but if you don't know, make sure you visit the placentation, the comparative placentation site. Here's the website. It has an amazing amount of information and literature review type stuff, but it also has some pretty amazing images that Dr. Benershka took. He was a human placenta guy, and then as he started working with the San Diego Zoo, he was fascinated by the diversity in animals. The sloth has a syndesmal chorial and an endothelial chorial. So syndesmal chorial is somewhere here between epithelial and endothelial chorial, and then endothelial chorial. The 9-banded armadillo is hemochorial. So there is a little bit of variability in the invasiveness of the placenta. This kind of nomenclature basically just says how much of the placenta on the mother's side disappears and is invaded. And it also gives you an idea of how important that drinking of colostrum is, because animals that have this kind of placentation where the mom's side also is protected by 3 layers, as well as the fetal side. They're the ones where the antibodies are hard to pass across the placenta, and so getting colostrum is absolutely of paramount importance. Whereas in primates, a lot more of the transfer of passive immunity antibodies is done through the placenta. Colostrum is still important and a good thing to have for many other reasons, in addition to the immunology one, but it's not as critical from the immune point of view as it is for some of the hoof stock. They are discoid as well. In the giant, and this is a southern tamando again. Here you can see the discoid part of the placenta. It's like that same image that I had earlier, and you can see it. In the two pregnancies that I followed, I did not appreciate any changes in the placenta, but knowing now if I had known then what I know now about placentation, it probably would have made a more concerted effort to get additional imaging. So go more frequently towards the end and actually image and be able to document any of these changes towards the end. In primates, you can detect like a letting go of the placenta, which really kind of spells out the impending birth. As far as mammary glands go, the anteaters and sloths tend to have one anterior pair, and some of the armadillos have thoracic and inguinal pairs, probably associated with a number of offspring that they have per pregnancy. I had mentioned to you that some of these births, they call them non-seasonal because they happen throughout the year, but you can see definitely peaks in, months in which it is much more common and months in which it's much less common for these births. There have been documented changes in the season of births in animals that are native to the southern hemisphere and then are, maintained under human care in the northern hemisphere. And so it is always important when you go through the literature or when you document stuff to spell out where you are so that it can be interpreted within that context. There are other species where it doesn't seem to really affect on which hemisphere they are, but and now I'm speaking of species in general, not just necessarily Xenarthrans, but there are a lot of species for which within the Xenarthrans we're missing some of this information. And I would just like to encourage anybody publishing on it that they, take a look at some of these things. And if you have those data, just provide them to the rest of us. Okay, so the reproductive parameters for the females. I couldn't find, again, if they documentation of true spontaneous versus induced ovulation. So some of them, there's also a lot of the cycle length missing because we don't have those samples often enough. And like I was mentioning, I have tables on sloths and anteaters as well. Gestations, we do have a little bit more information on these. Some of these are from observing them in the wild, not necessarily. So when, mating season is and then when the births are. Not necessarily because they were followed endocrinologically. There are a couple of these in which diapause has been documented, and then the litter size is probably one of the easier things to document, so that's one of the columns that is best populated. When you have a spontaneous ovulation, you will have a rise in progesterone following every estrogen peak, even if there is not a male. For the cycle length, you do need serial hormone data so that you can measure between the two estrogen peaks. Because if they do have an induced ovulation, you still can record interestrus seasons or interestrus periods, but they won't all be followed by a luteal phase. So it does mean that sometimes when you have, like in cats, you'll have short interestrus periods, but then if they do ovulate, it'll be a 2-month period between one and the next estrus. And so for these, you would need to really have accurate data. It would be good to measure it with hormones as well. So measure ovulation and then the period. So include progesterone if you're going to do that. And then for the diapause, what you would see is a rise in estrogen and then a delayed rise in progesterone. And there's other examples, for example, in otters where they have documented this. And I think it would be great to have some more of that kind of data, those kind of data for Xenarthrans. In the sloths, we have these two where it was monitor that it was spontaneous. I forget which one of these two authors, what they did was measure progesterone. And so in females that were not housed with males, if there is a rise in progesterone, it had to be a spontaneous ovulation. And then in some of these, they measured, you know, peaks in between the progesterones. And then gestation, same thing. Diapause has not been documented in any of the sloths. And then the litter size is 1 for all the ones that I was able to find. The giant, and then among the anteaters, ovulation has been documented to be spontaneous in these two. I think it's fairly safe to assume that the northern tamandra would also be spontaneous. Sometimes some of the more solitary animals can have, other species have a higher frequency of induced ovulators because it's not always easy to find a mate at the same time as one is in estrus. So it is handy to have it be induced, but if the density of the population is high enough, then there wouldn't be a need from an evolutionary point of view. And then here's some of the cycles that they have been able to measure. And then gestation from the estrogen peak to parturition, and 1 from breeding. So you can see that the ones from breeding can be a lot wider in their range because they may not necessarily have gotten pregnant on the 1 cycle, and then the second breeding wasn't observed. Or some animals, some species may breed even though they're pregnant. I don't know that has necessarily been documented in Xenarthrans yet. Diapause, there is some conflicting evidence about the anteater. I think there was 1 paper that says that it was, although I've heard other authors say they're not convinced as much about that. I don't have any personal experience or hard data to point one way or another. Also 1 per pregnancy, and it is rarely twins. Oftentimes they won't survive if it's twins, or they won't both survive. Ultrasound is one of those things that are considered ART, just mostly because it's using a little bit more technology. We did develop an ultrasound guided transcervical uterine biopsy technique. It was a technique that we were developing for primates, but given the anatomy of the tamandua and kind of what we were finding on ultrasound. So we were finding changes in ultrasound in the endometrium that we really kind of wanted to biopsy. So we decided to try the same technique that we were using in primates. So we used the endoscopy to have direct visualization of the tissue. And in primates, it was really useful to be sure that we were going through the cervix, because getting through the cervix is a little bit more challenging. It was super easy in the anteater, and after we discussed the anatomy, you know why. So we were able to visualize this and know where we were going into. And then with ultrasound, we had a visualization of where the endoscope was at so that we could be sure that we were sampling, that we were biopsying the parts that looked abnormal on ultrasound. Because oftentimes when you go endoscopically, if you can reach, say, the cervix, but then you have to put the biopsy instrument beyond the scope, you're not necessarily seeing what you're biopsying. So this allowed us to do both. And here's an image of the biopsy instrument with the ultrasound. So we're taking the ultrasound transabdominally, and the sampling is transcervically. You can get usable biopsies. They can be read out with the biopsy forceps. If you have the biopsy forceps, it allows to get multiple samples of different areas and make sure that you get a good representative sample of all the different lesions that you want. And in primates, it's really handy because you're not having to go through a narrow cervix. In the anteaters, it's not as big of a problem because it's not as a constricted space, but you're still, if you go through the channel, you're still diminishing a lot of the trauma that you might be causing by just passing the biopsy back and forth as you're trying to get it out of the animal and into your jar. We did document endometrial hyperplasia, and it was associated with infertility. This was that same anteater that we had already observed for 2 pregnancies in a row, and then she failed to get pregnant the 3rd time. And I was surprised a little bit like that because endometrial hyperplasia in other species tends to happen when the uterus is not, you know, when it lays vacant for a while. And that was not the case in this female. I was also contacted later on by another zoo, and they had another female Tamandua that had also endometrial hyperplasia. What we ended up doing in both of these, what we did in her and we recommended for the other one, was doing a hysterectomy, which we were pretty wary about because obviously with the clause that they have, the post-op care was going to be challenging and making sure that she didn't really pull out all her stitches, but she was actually really good. She left it alone. So the endoscope that we used was a Storz. It was a 1.9 millimeter with a 1.7 biopsy forceps. And you can see that one here. It's the kind that you put in through there and it's flexible and then you pass it through and then you can get it. So this was the setup. And then externally, we were using an ultrasound, and these are the two models that we happen to use. This one gets, the Fazone gets a really nice image, and the EI Medical one is super handy for travel, for moving it. And so this is kind of what it looks like going into the uterus. It was so light that I was at first thought, oh my God, did we end up in the bladder as opposed to the uterus? But you could see these are the blood vessels, and it was actually really light, and we got good samples, and we did not punch a hole in the bladder. It was not the bladder, so it was actually the endometrium. So we were a little bit worried about being in the right spot because it was so easy to get to. We kind of like fell into the uterus. You just advance it and you're there. So it's a really easy technique to use if you need to do this. And then ultrasonographically, we were confirming that we were in the area of the lesions that we wanted to biopsy and find out what it was. And so it did start bleeding a little bit once we started sampling, but she was fine and it wasn't excessive. And we got really, really good biopsies. So you can see, and you go to a different area and it's perfectly fine. So the only thing that when you do this is you want to warn them that they are going to bleed a little bit, but it shouldn't be excessive. And I think that's all that I had. So do you guys have any questions? And thanks again for the invitation. It's a pleasure to be here.
All right, so my name is Kenny Coogan. I'm part of the IUCN, International Union of Conservation of Nature, SSC, Anteater, Sloth, and Armadillo Specialist Group. And we host webinars along with some other educational programming. You can learn more by going to Xenarthrans.org. We have an Instagram page, we have a Facebook page, and those are only made possible with the help of our specialist group members and people who attend the webinar. So if you have photos of any of those animals, please send us a message on either Instagram or Facebook, and I would be happy to share them. And about several months ago, we started a YouTube page where we post our webinars as well as some other educational materials, which I'll show you in just a minute. If you go to our website, Xenarthrans.org, you will see that we have all three of the taxonomic families there. And then we have every species listed. And like Anneke was saying, there's a lot of armadillo species, and we have them all listed. All right, so if you go to our website or if you go to our Facebook or YouTube channel, you will see that we have several animated videos about the evolution of Xenarthrans, and then we have a special video on just armadillos, just sloths, and just anteaters. They're all about 5 minutes in length. We have them available in English, Spanish, and Portuguese. If you are educating children, adults about these animals, we encourage you to share this resource with them. Along with the animated videos, if you go to our website, education, and then you go to the Kids Corner, you will see that we have a lot of different activities based on those characters that are in those short animations. So we have word searches, we have coloring sheets, we have spot the differences, and just like the videos, we have them available in the 3 languages. Next month, we have tamanduas. We're talking about animal ambassadors. So that's a webinar. And then in June 14th, we will have Lindsay Sears, who will be talking about the Taxonomic Advisory Group and the Species Survival Plan programs about Xenarthrans. And like I mentioned, if you have photos of your animals without cages or bad backgrounds, or if you have Xenarthran jewelry or art that you want to share, we are happy to share that on our website. If you feel inspired to donate to our Xenarthran group, you can go to our website and click that donate button, and that will help with our education and our conservation efforts. And if you don't want to just give a straight out donation, you can go to our brand new store. It's a long title, Anteater, Sloth, and Armadillo.creator-spring.com. But you can order a super cool t-shirt like I'm wearing right now with a glyptodon, an ancient armadillo, or you can get a giant anteater. You can get the anteater family. You can get 2-toed and 3-toed sloths. You can get the giant armadillo. You can get hairy armadillos. We did about 12 different characters. We have the 9-banded with its 4 children, its 4 identical children, like Anneke was talking about. You have the glyptodon, and all of these come in like so many options. You have different colors. You can even get a shower curtain, a mug, a notebook. If you like Xenarthrans, we are here for you. And all of the proceeds benefit our specialist group. Other organizations who benefit our specialist group are FIA, the Foundation for International Aid to Animals, and Nurtured by Nature. Both of them support our education initiatives, which include creating those animated videos, and they also help us host our monthly webinars. All right, with that, we might have some questions for Dr. Anneke. I know you had a little video, or not a video, you had a little message from, I think, Argentina.
Yeah,
yes,
they've been asking some questions on the chat, and so while you were talking, I was answering some of those.
All right, so I'm going to read it because when we post it on YouTube, people cannot see that. So San Diego Zoo, for the tamandua spermatogenesis, you know, the creation of sperm, having rest and rut periods, do you know if this has only been observed in the wild or is it documented in tamanduas under human care? Any advice of how to monitor this?
So I can send you the papers that I was able to find on that. I think that if you are at San Diego and you have a male that you can monitor, I would definitely, you know, try ultrasounding them on a regular basis. They're very amenable to it, and it's not hard to find the testes. So you can measure the testes and you can see differences in there. The other thing that you can try to do that I've done for monitoring spermatogenesis in some animals, I mean, obviously the one way you could do it is to, you know, try to collect semen. But if the animals are not trained, you really probably don't want to be anesthetizing them just for this, you know, on a regular basis. If they let you, you can try to kind of swab the urethra opening, maybe with a small cotton-tipped applicator and then doing a smear. It's not a for sure thing because you'll only find it if they have ejaculated. But that is one way. If you really truly want to just find out for sure, there are studies out there where, you know, they anesthetize animals once a month and they check. I don't know that that would necessarily be worth it in general, but if it's something that you guys really need to find out, that would be one way to go about it.
Thank you for that question and answer. Adriana asks, hi, greetings from Costa Rica. We have educational tours about sloths, and the visitors are always surprised why the 2-toed sloths have a more prolonged pregnancy than 3-toed sloths. Any additional comments about that?
Yeah, so I didn't really look up for the age of sexual maturity, but I was just wondering when Adriana asked that question if it was maybe tied into how fast they mature in general. Like I had that same reaction when I was first learning about like hippos. They're about the size of a rhino, and yet the hippo pregnancy is 8 months and the rhino pregnancy is 16 months. So it's a huge difference, but I don't know enough about those two groups of species to to say that, but I think it would definitely be interesting to look at when they mature and if maybe their metabolism is a little different. I mean, they both have very low metabolism, but maybe one of them is even lower than the other one.
Mariella says that she studied the reproductive cycle of peaches for her PhD, and we didn't find regular ovarian cycles and actually suspect they are induced ovulators. It wouldn't surprise me if other armadillo species have the same strategy.
Yeah, I think that would be fantastic to document that. Did you run hormones on fecals, or how did you do that?
She may be unmuting herself. Yes, she is.
Yeah, here I am. Yes, we analyzed fecal hormones.
Oh, cool.
So we did estrogen and progesterone in females and corticosteroids.
Oh.
And in males, we also did hormone analysis. So we saw that the males definitely had seasonal reproduction. We also did histology, actually, based on confiscated animals. So the animals were confiscated dead, so we were able to extract ovaries and testes. So we did histological studies on one hand, and then we combined that with hormone studies in captive kept animals.
Cool. Yeah, I mean, definitely the cortisol might have affected some of the ovarian cycles. But cool. Yeah, I think it would be fantastic to really be able to document them being induced in other ones as well.
I mean, ecologically, it makes a lot of sense.
Yeah, absolutely. Absolutely. But without having the type of studies that you run, it's hard to tell. But I think that those would be really rewarding studies, because if you are suspecting that it isn't ecologically, it makes sense. It would be really cool just to have the hard data. So
yeah, we tried that for 4 or 5 years to determine what would induce ovulation. It was not that easy because you cannot take so many blood samples.
no.
From
And I mean,
female.
you know,
So.
there are some, for example, and there are some animals that are inducible, so they're not obligate induced animals, right? So like domestic cats that people think of as induced ovulators, they actually also from time to time ovulate spontaneously. Because if you have a whole bunch of females housed together, they will also ovulate, or if the male is next door. So it doesn't actually have to be copulation, but obviously with their penile spines, if they do copulate, they are induced, and they have documented that rise in LH. So those are also, I think, some of the ones that might be even trickier if they are facultative induced ovulators, but not always like that, because you are going to need a lot more data to prove that.
Yeah, that's a complicated part, I guess. I mean, it was interesting to see that we did not find any corpus luteum on any of the ovaries, except for 2 females that had aborted recently. So, you know, there are some other data that were really that led us to suspect that.
Yeah.
But,
and I mean, if you have somebody that is good enough with the ultrasound and have a good enough machine, I mean, even in the wild, you might be able to do ultrasonographic studies to look for corpora lutea.
Yeah, it's kind of tricky because peaches weigh 1 kilo and they're so susceptible to stress. I mean, we tried. I mean, I'm keeping them in my backyard. So I thought that they would be used to being handled, and they're so susceptible to stress that you cannot do an ultrasound.
Yeah, no, I meant like with some of the other species for which, I mean, you documented in these, but like in some of the other species where it hasn't been documented, maybe ultrasound, especially with the new technology that makes ultrasounds a lot smaller and more portable, it would be interesting to have more data from the field as well.
Yeah.
Cool.
All right, very good. I'll just follow up with San Diego Zoo Ambassador Keepers. They added that we can definitely try measuring our Tamanduas testes over ultrasound or swabbing after a breeding event. Thanks for the ideas.
Well, yeah, let me know how that goes. I'm curious.
All right, very good. And for those who are attending live, I put our store merch where you can buy a shower curtain all the way to a notepad with Xenarthran on it in the link. And if you're watching this on YouTube or Facebook, the link is in the description below. And thank you so much, Dr. Anneke Moresco, for that wonderful presentation. And everyone else, I hope you attend our next monthly webinar.
Thank you. Wonderful job doing the sales.
Thank you so much, Anneke.
Yes.
Thank you, Mariella. Hope to
Thank
meet
you,
you in
everyone.
person one time.
Hope so.
Bye,
Take care.
guys.